we'll transition now to Dave Glidden. So he's a colleague of ours who's worked in HIV prevention for more than a decade. As we know, many of us know him best as the lead statistician on the IPREX study, which was, of course, the groundbreaking study that first showed that oral Truvada is effective for HIV prevention as pre-exposure prophylaxis, and was involved in getting that intervention approved and licensed by the FDA, and subsequently has been really active in the field of novel trial designs for PrEP products in particular, and for HIV prevention in general, and thinking through how we can navigate this landscape and develop new trial designs. And I believe we'll talk about a Bayesian approach to leveraging external data for trial design. So Dave, take it away.
Thank you, Holly, and thank you, Sheila, for opening up those themes so very well that I will be talking about. And yes, this is a talk that represents a substantial collaboration between myself and some colleagues at University College London, as well as Gilead, and Peter Anderson, who taught me everything that I know about drug levels. I'm going to talk about something that Sheila alluded to, which is the possibility of using Tenofovir pharmacology to help to reconstruct a hypothetical placebo arm. I want to say I was first introduced to thinking about this topic by Rafi Landovitz, who I should definitely acknowledge, but it took me a while to think of a way that I could actually do this as I tried to find a way to advance my slides. Sheila was looking forward. I'm going to look backward, maybe giving us a hint on how to move forward. And I'm starting with the trial, the Discover trial, alluded to by Sheila, which compared Descovy or FTAF to Truvada or FTDF. It's the first trial of kind of a second generation, one that did in PrEP that did not have a arm that did not receive PrEP. It was a trial that enrolled almost 5,000 participants. It's a very large trial, twice as large as the IPREX trial, which was in a similar population, was placebo-controlled. And participants were asked to take 2 pills per day. They were taking an FTAF, either active or placebo, and an FTDF tablet, which was either oral or placebo. What's particularly interesting about a design like this is the lack of a group that gets no PrEP. I'm going to try to avoid the P word in referring to such a group, because even designs where people do get PrEP, there are sometimes placebos involved. So I'm imagining the idea, this is a trial that has no group that does not receive PrEP, for one. Second, it's a double-blind, double dummy design in which it's possible to really imagine a clear counterfactual scenario. And the clear counterfactual scenario is, I would like to imagine what the incidence had been if this trial had actually given 2 matching tablets and they were both placebos. Now, if we believe that the blinding in this trial was strong, we have no reason not to believe that, then that hypothetical scenario would apply to both arms. A second very important aspect of the Discover trial was that it employed a case cohort design for testing drug levels, and in particular, drug levels by dried blood spots. So the primary results of the Discover trial are that with over 8,000 person years of follow-up in each arm, the number of HIV infections is quite low. And I am in particular interested in the infections that appear to have occurred after randomization. It's a little bit ambiguous in the case of Discover, as they did not collect viral load samples at the enrollment visit, and we can't be 100% sure, but it appears that 5 participants were infected prior to randomization. That leaves us with 6 on FTAF and 11 on FTDF. And when you calculate the relative risk, that does clear their margin for showing that FTAF was non-inferior to FTDF. But the striking part of this trial is this very low rate of HIV infection in each arm. And in a future without clear no PrEP comparator groups, the question really becomes, what do these rates mean? Are they low because both products are effective, even potentially highly effective? And I mean that in the ITT sense in this trial. Or is it possible that the background HIV incidence rate, which is what I'm calling the situation that both groups get matching placebos, or is the background HIV incidence rate low or potentially very low? Now, there's a lot of circumstantial, there's a lot of external data besides the HIV seroconversions that we collect in such trials. Sexual practices, sexually transmitted infections, both of which point to potentially high risk in this population. But don't forget that background HIV incidence rate is also affected by treatment by as prevention. Another source of information is the case cohort design using dried blood spots. In particular, I want to look at the FTDF arm because that's where we have some data from prior studies that indicate what those levels mean. And this is work that's been done by Peter Anderson's lab, who's found useful categorizations of dried blood spots level, been able to map those to patterns of pill taking, less than 2, 2 to 3, or greater than 4 pills per week. And we're able in the Discover trial to ascribe HIV seroconversions post-enrollment to different patterns of pill taking. And in addition, because of the case cohort nature of the design, we're able to estimate the number of person years that those represent. You can begin to get the sense that that low category has something like a 4 per 100 person years type rate, and the rates are much, much lower as we get to the higher levels of pill taking in Discover. So I want to belabor also the fact that these 5 baseline HIV infections are themselves informative. They're people who are antibody negative at the enrollment visit. So you had to show that you were HIV negative at the enrollment visit, but they only did antibodies. And so the fact that these people screened negative earlier, screened negative at the enrollment visit by antibody, and then were positive at a subsequent visit with a viral load suggests that they're kind of a recent infection. And that's been one way that we've been talking about methods for inferring background incidence. And assuming that the mean duration of such a period is something like 14 days, it's possible to estimate person years associated with that and even ballpark a rate based on that snapshot of the trial participants just prior to their enrollment. And I mean that in a very metaphorical sense and also technical sense, because I'm estimating that rate using the Brookmeyer type snapshot estimator. That's really quite a wide boundary that we're seeing here, from 1 per 100 person years up to 6 per 100 person years. So what I'm interested in is the different ways that we can use external data around a trial to help to augment the HIV infections we see on a PrEP trial that may include 1 or hopefully 2 arms. And there have been a variety of methods that have been proposed using different kinds of external information to infer a counterfactual placebo rate. Now, across all of these, there are 2 things I think stand out as concerns. And I heard that a little bit in Holly's question. And that is each of these, we want to think about how does the information from an external data source relate to our particular trial population. So we're comparing across two different contexts, we're importing some information. And the second is that we're so aware that as we import across people, we're also importing across time. And whether or not methods that we use to inform our current trial could accommodate what we typically expect as decreasing HIV incidence due to treatment as prevention. But I'm particularly interested in this idea of using drug levels to reconstruct the placebo rate. So I'm going to lay a little bit of notation on you. The notation is fairly straightforward. I want you to remember that 2 will index FTAF and 1 will tend to index FTDF, and 0 will index the scenario without PrEP. And I'm imagining the number of failures that we're gonna, or HIV infections that we'll see in a trial or trial arm, the total follow-up that we'll see in a trial arm, in addition to thinking about those 3 substrata of DBS that I talked about earlier. And then the associated rate of HIV infections, which I'm going to treat as constant here, and often works well in our context where incidence tends to be on the lower side. I also want to introduce the efficacy associated with Truvada and with FTAF, as well as this rho sub L here, which is the proportion of total follow-up time on the FTDF arm, which is spent in the 3 strata of adherence, which I index with 0 up to 2 in increasing order of adherence. So, as I said, Rafi Landovitz planted this seed in my head about the idea of doing this kind of thing. But I spent a lot of time going around in circles about how it might be done. Because what I'm denoting here is what the stratum specific on FTDF rate of HIV infection might be. And here I've settled on a very, very simple model for that, where there's a factor for an intercept for even being on Truvada, and then I'm letting L be 0, 1, or 2. So assuming that the risk reduction across those 3 categories is identical from the lowest to the medium and the medium to the highest. But the issue was, of course, I had information that could inform those parameters. But what I was struck by, and you could even construct a Breslow type estimator for what the background incidence would be, is how to import the uncertainty that would come when I estimate those parameters beta 0 and beta 1 from historical data. So first of all, I knew this was going to be a difficult Taylor series expansion, very painstaking, as Faye can attest from her work on recency, propagating that error throughout things. But the thing that struck me was I was dealing with a case where the data in hand also was informative about the parameter B1, it's beta 1 itself. And that's when Bayes suddenly seemed very natural, because Bayes, when we think about it, if we have knowledge about parameters that comes from somewhere else, it's a natural framework for importing that knowledge through a prior distribution. It's also a natural way to pool data from different sources. It avoids complicated expressions as a requirement for propagating uncertainty throughout our inferences. So we can use simulations to do that for us. And it's also simple then to think about transformations of our different parameters. That's also easily handled through simulation. And it has some natural ways in which it admits sensitivity analyses as well. And I don't know how that happened. So I want to be very clear on what exactly I'm doing here. So there are three potential sources of data. And for each of those sources of data, we have to deal in Bayes with likelihoods and priors. So we have the FTEF data, which I'm going to use a straightforward exponential log likelihood. With Bayes, in for a dime, in for a dollar. I'd love to just have priors on the parameters, which are going to help me infer the drug levels. But if I'm going to use Bayes, I have to have priors on all my parameters. So for FTEF, I'm going to use a non-informative prior for its lambda 2. For FTDF, this becomes a mixture of 3 exponentials with the parameters that govern the risk reduction, and in addition, lambda 0 and beta 0, beta 1 now parameterize the log likelihood information for FTDF. We're no longer expressing it as its aggregate, we're expressing it as its component parts. And here I'm going to use previous studies to give me a prior for the beta values that relate the FTAF seroconversion rate to the background rate. And in addition, as in the case of having those baseline infections, I can also potentially augment my method with a log likelihood contribution from the recent infections, which would directly inform lambda 0, not just through beta. And also lambda 0 itself is something that I have to have a prior on. And I have 2 possibilities there. One is a flat prior, and the other is a variety of non-informative priors. And I considered one informative prior, which was centered at 0.5 per 100 person years. And that's to represent a sensitivity analysis in which we're thinking about the possibility that we believe that that background rate is quite low in this population because of, for instance, treatment as prevention. So I developed the prior based on data from the IPREX and IPREX-OLE data. The IPREX study was a randomized placebo-controlled trial of Truvada. We had a background incidence there of 3.9 per 100 person years. Now that study doesn't have concurrent dried blood spot measurements. Those were developed later by the lab of Peter Anderson, the same lab and same assay, which is being used for the Discover trial. So I have that information from IPREX-OLE, and what I did was combined the data from the IPREX placebo arm serving as a comparator and the IPREX-OLE case cohort arm in which I have DBS drug levels. And I formed a model in which I assumed a constant hazard for both periods that was shared, and I modified it by adjusting for some covariates and then also by the drug levels. And so the covariates that were used to adjust this comparison were age, the diagnosis of syphilis, conjugal intercourse, number of partners, and the study site. And I used GEE to correct for the fact that some people appear in both periods. So then once I estimate that beta, I take as my prior a bivariate normal distribution with beta hat 0 and beta hat 1 that were my estimators, as the center of that, and the variance covariance matrix as the variance covariance matrix of the bivariate normal. So I directly turn the estimators into a distribution for the prior. Run into a couple of, for as simple as my models are, you run into some complications pretty quickly. The first is that the DBS, and I'm talking about Discover here, are sampled by case cohort. And because Bayesian methods are likelihood-based methods, I have to appeal to the work of people who have developed the likelihoods for case cohort designs. It turns out that for people who the cases, the cohort have easy likelihood contribution, the non-cohort members have a more complicated contribution to the likelihood that does involve the parameters of the drug level, and that's that row 1, row 2, and row 3, the fraction of the risk period that is spent in each period. And actually, I'm working with aggregate data here because Gilead and I do not have a data sharing agreement. So actually, the exact likelihood contributions have to be approximated. The second issue that we've run into is that the rate for the FTDF arm is no longer exponential. We're interested in comparing that rate, for instance, to an inferred placebo rate, but lambda 1, which we're interested in, is no longer a parameter in our model. And it has a mixture distribution, as I said before, and we'd really like to talk about lambda 1. So I developed an expression for that approximates its least false value. And that's what I estimate from my parameters to reconstruct an overall estimate for the FTDF arm. And we see, for instance, that when I look at the posterior mean or median, that agrees closely with what was observed in the data. I did my work in STAN, which implements Hamiltonian Monte Carlo, drawing 20,000 draws from the posterior distribution. STAN is pretty amazing. It's pretty flexible. There are some things that would normally be hard that are very easy to tweak in a program like this. But it is really hard to get it to compile on your computer. It gives incredible flexibility in likelihoods, allowing me to implement this case cohort likelihood, and has many built-in capabilities. I want to give you some sense of the results that I get. So I'm looking back to look forward. So here I take the case that I do augment the likelihood with the 5 recent infections. I'm assuming a non-informative prior on lambda naught. And these are the results that I get. I get posterior medians. I get a back calculated posterior incidence for the background HIV rate of about 3.1 per 100 person years. You can imagine if you compare that to 0.25 and 0.14 infections per 100 person years as observed on Discover, both arms look highly effective. And here I'm able to formalize this by getting the posterior distributions, what their center is, and what the 95% credible interval is. And this is consistent with very high ITT effectiveness on both arms. There's a real issue here of the possibility of unmeasured confounding. And to address that, I contemplated the idea that the background incidence across the drug level strata may itself vary, okay? And allowed for the possibility across 3 drug level strata that the background HIV incidence varies. And I introduced this parameter phi, which is e to the phi is a ratio of how much higher the background incidence gets as the degree of adherence decreases. So I kind of build in this parameter that can index a degree of confounding. And I used a value of phi equal to log 3, which would imply that the group with the lowest drug levels has a background incidence which is 9 times higher than those with the highest adherence, even in the absence of PrEP itself. And when I implement that as a sensitivity analysis, here I'm using, again, a non-informative prior, a lambda 0, augmenting with those baseline infections, and I have phi equal to 3. We see that decreases the efficacy quite a bit because the background HIV incidence rate is now inferred to be 1.6. And so we ascribe much less of the decrease in HIV incidence to the higher adherence to or higher DBS levels. So I am trying to motivate two things here. One is I think that the use of drug levels has real value. It's going to be a contemporaneous control group, and we do have good assays which have in general linked very well to HIV risk reductions in previous trials. However, it is susceptible to confounding, but I hope I've convinced you that that is something that can be handled in sensitivity analyses. But the other thing that I'm trying to motivate is the value of Bayes for problems like this. The idea here is we'd have a likelihood for the trial data, and maybe we're augmenting that likelihood, possibly through prior distributions, possibly through other likelihood contributions, to allow for the other external data that we have. And that external data has in many cases been related to background HIV incidence in other trials. And I really want to kind of motivate that, especially because it's a method that can possibly synthesize data from many different sources. And Bayes is natural for these kinds of problems. It seamlessly propagates uncertainty. It allows us to deal with that uncertainty, not through analytics, but instead through Markov chain Monte Carlo. And I believe that we could possibly triangulate many different adherence modalities into this. And a final slide that, just if Holly will indulge me, I just wanted to stress that we can not only look at the efficacy relationship in each of the arms, but we can look at meta parameters that compare efficacy between the two arms. And on the right here, I have the ratio of those efficacies, also known as the averted infection ratio. And you see how tight the inference is here between 0.9 and 1.1, indicating substantial similarity in the case that I use a non-informative prior for lambda 0. And also I derive the difference in efficacies for those of you involved in SARS-CoV-2 vaccine trials. You know that, for instance, the FDA has talked about a 10% difference in efficacies as evidence of non-inferiority, and you would see that almost all the posterior lies within that plus or minus 10%. Thank you very much.